Welcome, everyone. I want you to just look down at your hands for a second. You might think you're just sitting perfectly still right now, but inside, you are a buzzing, hyperactive city of molecular machinery. Today, in this explainer, we're diving into Chapter 5, Life Processes. Let's uncover the invisible maintenance crew that is literally keeping you alive this very second.
If you see a dog running around, or even if that dog is just asleep on a rug, we know it's alive because we can see it breathing, right? But if you ask a professional biologist what truly defines life, they won't just look at breathing. They'll tell you that true life actually comes down to microscopic movements that our naked eyes can't even see. It's a nonstop microscopic hustle.
You see, our environment is constantly trying to break things down. Because of the chaotic effects of the outside world, the highly organized, ordered nature of living structures is always at risk of falling apart. To fight this, organisms act like a continuous maintenance crew. We must constantly repair and maintain our structures, and that requires moving molecules around all the time, 24/7. Here's our roadmap for today. We'll be looking at 1. The invisible molecular dance. 2. Plants, solar-powered factories. 3. Proving the recipe. 4. Heterotrophs getting our food. 5. Digestion in action. And finally, 6. Respiration unlocking energy. Let's get into it. Section 1. The invisible molecular dance. To maintain all this order and prevent our cells from just falling apart, we absolutely must import energy from the outside world. This process of transferring a source of energy from the outside to the inside is what we commonly call nutrition. Maintenance processes prevent damage, but they strictly require this constant supply. And hey, if an organism wants to grow, it needs even more raw materials. But where exactly do we get this energy? Section 2. Plants, solar-powered factories. Think of green plants and certain bacteria as incredible microscopic solar-powered factories. They use a process called Autotrophic Nutrition. They pull in incredibly simple inorganic materials from their environment, specifically carbon dioxide from the air and water from the soil, and they manufacture complex energy-rich carbon molecules called carbohydrates using sunlight and chlorophyll. So, how do they actually pull this off? Well, it happens in 3 distinct steps during photosynthesis. First, imagine the green pigment, chlorophyll, acting like a biological solar panel, absorbing the light energy. Second, that light energy is converted into chemical energy, and it's used as a microscopic blade to slice water molecules apart into hydrogen and oxygen. Finally, those pieces are used to reduce carbon dioxide, essentially baking it into a carbohydrate cake. Whatever energy the plant doesn't use right away, it simply stores away as starch for a rainy day. Section 3. Proving the recipe. Now we don't just have to take the textbook's word for it. In Activity 5.1, we take a potted plant with variegated leaves, meaning leaves that are partly green and partly white. If we want to test where the plant stored its energy, its starch, we first dip the leaf in boiling water, and then we carefully boil it in alcohol using a water bath. That water bath is super important so the alcohol doesn't catch fire. Why do this? Well, imagine trying to see a dark blue ink stain on a dark green shirt. It's nearly impossible. The alcohol strips away the green chlorophyll, turning the leaf pale. Then, we drop a dilute iodine solution onto it. The magic here is that iodine instantly turns blue-black the moment it touches starch. And brilliantly, only the areas that were originally green turn blue-black. We have visually proven that chlorophyll is absolutely essential to manufacture starch. And this perfectly sets up Activity 5.2. Here, we take 2 healthy plants and lock them each under airtight glass bell jars. But in one jar, we place a little dish of potassium hydroxide. Think of potassium hydroxide as a relentless chemical sponge. It actively sucks all the carbon dioxide out of the air inside that jar. After leaving them both in the sun, we do our iodine test again. The plant locked in with the potassium hydroxide shows absolutely no starch. Why? Because we stole its carbon dioxide. This proves for sure that without CO2, the plant's solar factory completely shuts down. Section 4. Heterotrophs. Getting our food. But what if you aren't a plant? Heterotrophs rely directly or indirectly on autotrophs for survival. Depending on whether your food is stationary, like grass, or trying to run away from you, like a deer, organisms have evolved wildly different strategies to get their nutrition. Let's strip it back to a single-celled organism, the amoeba. Since a single-celled organism's entire surface is in contact with its environment, it doesn't even need a mouth. Imagine the amoeba sensing a piece of food. It physically pushes its cell surface out into temporary shape-shifting fingers called pseudopodia. These pseudopodia reach out, wrap around the food, and fuse together, swallowing it whole into a tiny bubble called a food vacuole. Inside that bubble, biological scissors break the complex food down into simple nutrients that diffuse right into the cell, and any leftover trash is just pushed back out. Pretty amazing, right? Section 5. Digestion in action.
Because humans are incredibly complex, a simple cell surface just won't work for us. Oh, and a quick side note before food even goes down. Remember that bacteria in our mouths can act on sugars to produce acids? These acids literally soften our tooth enamel and cause dental caries, which is exactly why brushing your teeth is so absolutely crucial. Once past the mouth, we have a dedicated transportation system, the alimentary canal. It's a highly specialized, long tube extending all the way from the mouth to the anus. Each section of this tube has a very distinct job to break down complex carbon sources using incredible biocatalysts called enzymes.
Let me show you exactly how powerful these enzymes are with Activity 5.3. Imagine two test tubes, A and B, both filled with a starchy solution. In tube A, we add a little bit of our own human saliva. We wait about 20 minutes, and then we add our trusty starch-detecting iodine to both tubes. Tube B turns a deep blue-black, meaning the starch is still there, but tube A, no color change at all. The saliva contains a specific digestive enzyme called salivary amylase that acts like a microscopic chainsaw, chopping the giant, complex starch molecules down into simple sugars right there in the tube. Digestion literally begins the second food enters your mouth.
Section 6. Respiration. Unlocking energy.
Okay, so we've eaten the food and our digestive enzymes have chopped it up into simple sugars. But how do we ignite that fuel to actually power the molecular dance of life? Digestion is just the delivery service. It gets the fuel into the body. Respiration is the engine. It is the oxidizing, reducing chemical reaction that actually burns the fuel. We must acquire oxygen from the outside world and smash it into our food to release the energy locked inside. Let's sketch this out on a mental whiteboard, kind of like looking at atoms through a really strong magnifying glass. Here's the chemical equation for respiration. You have a complex glucose molecule from your food, C6H12O6, and 6 oxygen molecules, O2, from the air you breathe. When they collide in your cells, they don't just disappear. The law of conservation of mass tells us that atoms cannot be created or destroyed, only rearranged. Think of them like Lego bricks. We take the glucose and oxygen Lego structures, snap every single piece apart, and rebuild them. The exact same number of carbon, hydrogen, and oxygen atoms that went in must come out on the other side. They get rebuilt into 6 molecules of carbon dioxide, 6 molecules of water, and in the process of snapping those chemical bonds, pure energy is released. This energy is immediately used to synthesize a molecule called ATP, the universal energy currency that powers every single reaction in your cells. And we can prove that carbon dioxide is the exhaust gas of this chemical reaction right now using Activity 5.4. We take freshly prepared lime water, which is a totally clear liquid. If you take a syringe and pump normal atmospheric air into it, it takes a very long time for anything to happen. But if you take a straw and blow the exhaust air from your own lungs directly into the lime water, it turns a cloudy, milky white very rapidly. That milky color is the direct chemical signature of high concentrations of carbon dioxide. You are literally watching the exhaust of your cellular engines. Now, while humans use oxygen in this process, which we call aerobic respiration, Activity 5.5 shows us that lower organisms have a neat trick. If you put yeast into some fruit juice and block out the oxygen, the yeast will still break down the sugars to get energy. This anaerobic process ferments the juice, and it still produces a gas. If we pipe that gas into lime water, once again it turns milky. Even without oxygen, the yeast is producing carbon dioxide as it desperately burns fuel to stay alive. And hey, speaking of oxygen, this is exactly why aquatic animals like fishes, which you can observe in Activity 5.6, breathe much faster than we do. There is simply less dissolved oxygen in water than in the air we breathe, so they have to work harder to get it. So we've broken down our food and we've seen the chemical fire that burns it with oxygen to create ATP energy and carbon dioxide exhaust. But here is the massive logistical challenge. If you are a multicellular human being, how on earth do you transport that specific sugar molecule and that specific oxygen molecule from your stomach and lungs all the way to a microscopic cell in the tip of your little toe? And how do you get the toxic waste back out? That incredible transportation system is exactly the mystery we'll be exploring in our next explainer. Keep wondering, keep learning, and I'll catch you next time. All right, let's just dive right into this. Welcome to this explainer, where we are going to take a journey inside the absolute most bustling, complex metropolises on Earth. And no, I'm not talking about New York or Tokyo. I'm actually talking about the living bodies of plants and animals. Today, we're going strictly ground up to discover exactly how these living cities deliver fuel to their residents, and, just as importantly, how they manage and remove their toxic waste. We have to start with the ultimate biological engineering problem. I mean, really think about this for a second. The human body is a massive city of roughly 30 trillion individual cells. How in the world does every single one of those microscopic residents get a continuous, nonstop supply of oxygen and nutrients to survive without a single delay? Because, well, if the delivery trucks stop, the cell literally dies. So here is our roadmap for today. 1. The human pumping station. 2. Blood, vessels, and lymph. 3. Plant transport, moving water and food. And 4. The waste removal system. Okay, let's hit section 1, the human pumping station. This is our incredibly powerful dual-action engine.
Imagine zooming in on this organ with a magnifying glass. Your heart is roughly the size of your closed fist. But notice, it isn't just one big hollow balloon. It's highly structured, carefully divided into 4 distinct rooms, or chambers. Now, why all the walls? Because our body absolutely has to prevent oxygen-rich, fresh blood from ever mixing with the carbon dioxide-rich, dirty blood. It's kind of like keeping your clean drinking water pipes completely separated from your sewer lines.
Let's trace this double circulation step by step in our minds, like we're drawing on a whiteboard. Fresh, oxygen-rich blood from the lungs arrives at the thin-walled upper left chamber, the left atrium. The atrium relaxes to collect the blood. Then it contracts, dropping it down into the left ventricle. Now, the left ventricle has these incredibly thick, muscular walls. When it contracts, it squeezes with massive force, pumping that oxygenated blood out to your entire body. Meanwhile, the used, deoxygenated blood returns from the body into the upper right atrium, drops into the right ventricle, and gets pumped right back to the lungs to pick up fresh oxygen. It's 2 completely separate loops happening simultaneously.
So you might be wondering, why do we need this strict 4-chambered separation anyway? I mean, fishes only have a 2-chambered heart that pumps blood in a single, simple loop. Amphibians have 3 chambers, so they tolerate a little bit of mixing. But birds and mammals, like us, we are warm-blooded. We have extremely high energy needs because we are constantly burning energy just to maintain our internal body temperature. If our fresh and used blood mixed, our oxygen delivery would become terribly inefficient, and our cellular engines would basically just stall out.
Moving on to section 2, blood, vessels, and lymph. These are the highways and the sanitation workers of the body. Our blood vessels act as specialized highways, and trust me, they are not all built the same. Arteries are your high-pressure outbound roads. Because the heart is violently squeezing blood away into these tubes, arteries need thick, super elastic walls to handle that massive force without bursting. Veins, on the other hand, are the low-pressure return streets collecting blood to bring it back towards the heart. Because the pressure is mostly gone by the time the blood gets there, veins have thinner walls, but they have a really cool special feature: one-way valves. These act exactly like toll gates to stop blood from flowing backwards due to gravity. Now, the physical force pushing through these arterial highwinds is your blood pressure. We actually measure this with an instrument called a sphygmomanometer. When that left ventricle intensely contracts, the pressure spikes. This is your systolic pressure, normally around 120 millimeters of mercury. When the ventricle relaxes to refill, the pressure drops. That's your diastolic pressure, normally around 80. So you know when the doctor says your blood pressure is 120 over 80? Well, this exact mechanical pumping cycle is literally what they are measuring. But what happens if this high-pressure plumbing system suddenly develops a leak? Like, if you get a cut, you start bleeding, right? If you lose too much blood, the physical pressure of the entire system drops, which would be catastrophic. Enter the platelets. These are specialized blood cells that act as your body's emergency plumbing repair crew. The second a pipe breaks, they rush to the site of the injury and immediately plug the leak by clotting the blood, sealing the whole system, and saving your life. This brilliantly illustrates the complexity of our fluids, but we definitely can't forget our secondary transport system, lymph. While blood is red and packed with high protein to transport oxygen, lymph is a colorless tissue fluid containing way less protein. It basically escapes through the pores of your capillaries into the spaces between your cells. Lymph has entirely different jobs. Its key function is actually carrying digested and absorbed fats away from your intestines and draining excess fluid back into the blood. All right, section 3,
Plant transport. Let's see how we move water and food inside the botanical skyscraper.
Think about this absolute engineering marvel for a moment. Plants don't move around, and a lot of their tissues are made of dead cells, so their overall energy needs are pretty low. But the distances they have to transport materials, they can be massive. How on earth does a 100-foot-tall tree pull heavy water entirely against the force of gravity, from the deep soil all the way up to its highest canopy, without any muscular pump or heart at all? To really understand this, let's play detective for a second and explicitly walk through Activity 5.8 from your textbook, the transpiration mystery. Imagine you take two identical pots of soil. In one, you have a living plant, in the other, just a lifeless stick. First, you cover the top of the soil in both pots with plastic so absolutely no water can evaporate directly from the dirt. Then, you cover both entire setups, the plant and the stick, with large, clear plastic sheets and leave them outside in bright sunlight for about half an hour. What do you actually observe? Well, water droplets rapidly form on the inside of the plastic covering the plant, but not the stick. So, what does this prove? It proves that the aerial parts of the plant, specifically the leaves, are constantly losing invisible water vapor right into the air. This crucial scientific process is called transpiration. As those water molecules evaporate and exit through the tiny pores in the leaves, the stomata, they create a real physical suction force. Because water molecules kind of like to stick together, losing a drop at the very top acts like a giant invisible vacuum, continuously sucking up a solid column of water and dissolved minerals straight up from the roots. The tree is literally drinking through a giant straw using the heat of the sun. How cool is that? So, plants essentially have two independent transport pathways. The xylem is the water pipe. It moves water and minerals upwards using those simple physical suction forces from transpiration. But the phloem, which transports the food and sugars produced during photosynthesis, is totally different. This process is called translocation. Unlike the passive xylem, the phloem is like a premium delivery service. The plant actually has to spend real, cellular ATP energy to actively pack sucrose into the phloem tissue. That creates an osmotic pressure that pushes the food up or down to wherever the plant needs it to grow. And finally, Section 4, the waste removal system. Welcome to the sanitation department. With all this food and energy being consumed, toxic waste inevitably build up. Let's look at the human excretory system. Your lungs already handle removing carbon dioxide gas, but liquid, nitrogenous waste is handled by a pair of bean-shaped organs, your kidneys. The blood flows into the kidneys to be deeply filtered. And hey, if the kidneys ever fail, doctors use an artificial kidney process called hemodialysis to do this life-saving filtration mechanically. Normally, though, the resulting toxic liquid waste, which is urine, is sent down tubes called ureters to be safely stored in the urinary bladder until it's released through the urethra. But the real magic is happening at the microscopic scale inside those kidneys. Imagine zooming in millions of times into the kidney tissue. You would find tiny, specialized, cup-shaped filtration units called nephrons. These are the unsung heroes of your body, the microscopic cleanup crew. As high-pressure blood forces its way through the capillaries here, the nephrons expertly pull out those dangerous soluble nitrogen compounds while carefully reabsorbing the good stuff like glucose, amino acids, and vital water right back into your system. Wow, we have successfully navigated the bustling circulatory highways of the human body. We've scaled botanical skyscrapers using transpiration suction, and we've watched microscopic nephrons clean our bodily streets. And as a quick side note, plants do their part too by simply shedding dead leaves or storing their waste in cellular vacuoles and old xylem resins. Before we wrap up this explainer, I want to leave you with this absolutely crucial textbook question to test your new conceptual mastery. Knowing what you now know about warm-blooded energy needs, cellular fuel delivery, and the intense design of our four-chambered pumping station, why exactly is double circulation an absolute necessity for human beings? Think back to the huge difference between our energy needs and those of a simple fish. Keep exploring, keep visualizing these incredible life processes, and I will catch you in the next explainer. All right, let's get right into it. If you're a 10th standard student gearing up for those highly competitive Karnataka SSLC board exams, specifically subject code 83E, you are in the absolute right place for this explainer. Look, I know you already know the basic biology, right? You know what a heart does, you know how plants breathe. But today, we're not just passively studying biology. We are putting on the hat of a veteran board evaluator to get you the exact marks you've actually earned. We're going to dig into the official key answers and uncover exactly where the points are hiding in Chapter 6, Life Processes. So, Here's our SSLC exam blueprint for today. We'll be hitting the evaluator's blueprint, respiration comparisons, plant transport traps, the massive 4-mark heart diagram, excretion half marks, and finally, your final exam checklist. Let's do this. Section 1, the evaluator's blueprint. Time to start thinking like the person holding the red pen.
Okay, we really need a crucial mindset shift before we even glance at a question. Just knowing the science, it honestly isn't enough if you don't know what the board's marking scheme specifically demands. So hear me loud and clear on this. Stop writing massive, sprawling paragraphs. Evaluators are literally grading hundreds of papers. They do not have time to read an essay. They are hunting, just scanning your answers for specific keywords and formulas. Today, we're decoding those official SSLC key answers so I can show you exactly where the half marks and full marks live, and more importantly, where students just unnecessarily lose them. Moving on to Section 2, Respiration High-Yield Comparisons. These are the 3-mark and 2-mark absolute favorites.
So, looking at this exact table format, this is precisely what evaluators want to see for a 3-mark comparison between aerobic and anaerobic respiration. But wait, here is a massive exam trap based straight off the official A3E key answers. If you just write down this table, you're probably only getting 2 marks. Why? Because the official key strictly mandates that you must write the initial step first. You have to write glucose of 6-carbon molecule breaks down into 3-carbon molecule pyruvate. Simply writing that one sentence about pyruvate instantly secures your very first full mark. Then, writing any 2 differences from this table gets you the remaining 1 + 1 marks. So always start with that pyruvate breakdown.
Let's look at another super frequent tumor question. They'll ask, why is the rate of respiration more in humans compared to fish? You can't just write, well, fish live in water. No way, that won't cut it. The evaluator is hunting for 2 specific anchor phrases. For fish or aquatic organisms, you must write that the amount of dissolved oxygen is low in water. Boom, that's 1 mark. For humans or terrestrial organisms, you must state that the amount of atmospheric oxygen is high, and that's your 2nd mark. Low dissolved oxygen versus high atmospheric oxygen. Just memorize that direct contrast.
Section 3, Plant Transport Mark Traps. This is all about nailing those tricky 3-mark sequence mechanisms.
If they ask you how food is transported in plants, presenting your answer exactly as a 3-step sequence is going to perfectly mirror their official grading rubric. Step 1. Material like sucrose is transferred into phloem tissue using energy from ATP. Getting that sentence down, that's worth 1 full mark. Step 2. Osmotic pressure of the tissue increases and water moves inside. That snags you 1/2 mark. Step 3. This pressure moves the material in the phloem to tissues which have less pressure. There's your final 1/2 mark. So 1 + 1/2 + 1/2 equals your perfect 3 marks.
And this perfectly illustrates our most common mark traps. If you don't actually write the word ATP in that very first step, you will immediately lose a full mark. It honestly doesn't matter how beautifully you describe the rest of the process. If you forget the exact phrase osmotic pressure, poof, there goes another 1/2 mark. And of course, the ultimate, most classic trap, confusing xylem, which is strictly for water, with phloem, which is for food. Phloem transports food using ATP. Lock that right into your memory banks.
Section 4, the 4-mark heart diagram. The undisputed heavyweight champion of the biology exam. We have officially arrived at the ultimate big-ticket question for the life processes chapter. I'm talking about this schematic sectional view of the human heart. This is constantly a 4-mark question, and you absolutely have to master it. Look very closely at this official key from the 83E board materials. A massive 3 marks are awarded just for drawing an accurate diagram. The final single mark is split perfectly into 1/2 marks for labeling 2 specific parts that they request in the question, which in this case were the septum and the right atrium. 3 marks just for the drawing means your proportions and lines really need to be confident and clear.
But please, do not fall into these diagram mark traps. Evaluators check for these literally the second they look at your paper. First trap, missing or backwards blood flow arrows. Your arrows absolutely must show oxygenated and deoxygenated blood moving in the right directions. Second, loosely pointing your label line. If you're asked to label the septum, your line needs to actually touch the dividing wall clearly. And third, left-right confusion. Remember, the right side of the heart is drawn on the left side of your paper. Always double-check your orientation before you finalize your drawing in ink. All right, Section 5, excretion. Easy 1/2 marks. Really, don't leave these easy points on the table.
While the kidney nephron gets basically all the attention from students, the plant excretion section is actually a total goldmine for quick 1-mark grabs, provided you memorize the exact textbook phrasing the evaluators are hunting for. When they ask about how plants get rid of excretory products, do not just casually write, they drop their leaves. No, the key answer specifically allocates 1 full mark for this exact sentence. Waste products are stored as resins and gums in old xylems of plants. Resins, gums, old xylems. Those 3 terms are the magic keys to unlocking that point. Just don't leave easy points on the table by being vague here.
And finally, Section 6, the final exam checklist. Let's lock in your 100 out of 100 strategy.
Okay, we've covered a really massive amount of high-yield ground today. Let's just do a rapid-fire recall to lock these strategies in so you're totally prepared when you sit down in that exam hall. Keep this checklist burned into your mind the moment you open that exam paper. If you see aerobic versus anaerobic, draw a table, for sure, but write that pyruvate breakdown step first. If you see phloem transport, your brain should immediately scream ATP and osmotic pressure. And when you see the heart diagram, take a deep breath, draw it cleanly to secure those 3 base marks, and rigorously double-check your left and right labels. These right here are your non-negotiable points for maximizing your score.
You now possess the actual evaluator's blueprint for the life processes chapter. You know exactly what they want, you know how they grade, and you know the hidden traps that catch 90% of students off guard. So the question really isn't whether you know the biology anymore. The question is, the next time you sit down with a practice paper, are you going to write like a student, or are you going to write like the person holding the red pen? Good luck.